So DNA polymerization event is clear for us, like how DNA polymerization is done naturally by DNA, and we know that DNA polymerization process is carried naturally due to the pyrophosphate that is produced. The hydrolysis of pyrophosphate drives the reaction forward. And we also know that if there is only one phosphate left, DNA polymerase cannot seal it. It's called the nick. We need ligase to add an extra phosphate from ATP and then can seal the nick. Now we are going to talk about the different types of DNA polymerase. Different types of DNA polymerase that are available, and in prokaryotic replication particularly, we'll talk about different types of polymerases. So here you can see the table. In this table we have DNA polymerase. We have three different types of activities of a DNA polymerase and ultimately the function of that DNA polymerase. Now a DNA polymerase, as I mentioned, it has two kinds of activity. One is the polymerization, and the second one is exonuclease activity or proofreading, right? Or proofreading activity. Now polymerization direction is 5' to 3' polymerization is done by all kinds of DNA polymerase. But the exonuclease activity can be of two different types, 5' to 3' or 3' to 5' exonuclease activity. But not all the polymerases share same sort of exonuclease activity. Different polymerases have different types of exonuclease activity. Here we are seeing 5 different types of polymerase. So polymerase 1. Polymerase activity for all the polymerases are 5' to 3'. But for polymerase 1, the exonuclease activity 5 to 3' exonuclease activity is present. 3 to 5' exonuclease activity are also present. So that means DNA polymerase 1 can go 5' to 3' polymerization, and it can also go exonuclease activity in both directions. In both the directions. That is a unique feature of DNA polymerase 1. Unique feature of having 5' to 3' exonuclease activity, which is not present in any of other DNA polymerase. Well, the rest of the other DNA polymerase, like DNA polymerase 2 and 3, they have 3' to 5' exonuclease activities. Okay? So what are the function of DNA polymerase 1? Removes and replaces primers at the end of DNA replication event. Due to the capability of cleaving a DNA strand 3 to 5' or 5 to 3', they are excellent choice to utilize them to remove the RNA or any ribonucleotides from the existing DNA and fill that gap. Now what is a gap? Let's say this is the DNA strand. So these are hydrogen bonds in the middle, but here are two nucleotides, let's say A and G. No other nucleotide present in the complementary DNA. This region is known as a gap. So gap means nucleotides are missing. Gap means nucleotide are missing. Where a nick is where nucleotides are present, only the phosphodiester bond missing. This is the difference between a nick and a gap. So if there is a gap, what do you need to fill it? Nucleotides and polymerase. If there is a nick, then what do we need to fill it? DNA ligase is needed. No nucleotides are needed. And ATP is needed because ligase requires ATP to function. Clear? So the function of polymerase 2. Polymerase 2 is involved in DNA repair and it restarts the replication after damaged DNA halts in the synthesis process. And the polymerase 3 is the actual polymerase required for elongation of the DNA or DNA replication process in prokaryotes. So in prokaryotic DNA polymerase type, DNA polymerase 3 is the predominant DNA polymerase for the process of replication. And in prokaryotes, Polymerase 1 is used to fill the gap. Nix never can be sealed by polymerase. It can only be sealed by ligase with ATP. And polymerase 2 is used as a repair mechanism, a polymerase for DNA repair mechanism. Apart from that, there are DNA polymerase 4 and 5. 4 and 5, both this kind of DNA polymerase are used very less, but they are mostly used for DNA repair mechanism. Okay? And repair mechanism where translation means if there is a lesion formed in the DNA. Lesion means where there is no proper interaction or pairing of the nucleotides. A swell structure in the DNA strand. Those lesions can be removed and new DNA can be synthesized in that position by polymerase type 5. But mainly there are the three, Pol 1, Pol 2 and Pol 3. The unique feature about DNA polymerase 1 is that it carries a 5' 3' exonuclease activity.
Why is it necessary to remove the primers? Why don't the ligase ligate all with the primers?
বুঝলাম না বলো
Ligase the primerটাকে কেন DNA এর সঙ্গে মানে ligate করা যায়?
প্রাইমার তো তৈরি হয় আরএনএ কন্টেন্ট দিয়ে
কমপ্লিমেন্টারি
হ্যাঁ
এর সঙ্গে কমপ্লিমেন্টারি ওটাকে সরানো
তার কারণ হচ্ছে আমি ডিএনএ তে আর এনটিপি কেন থাকবে ডিএনটিপি থাকবে এনটিপি তো থাকবে না ডিএনএ এর মধ্যে যদি এনটিপি ক্যারি হয় তাহলে তো ইনফরমেশন থাকছে না ভুল ডিএনএ শুড ক্যারি 4 লেটার্স অনলি এজিটিসি নট এইউটিসি তো আরএনএ ফ্র্যাগমেন্ট কোনটা আসবে না কোনো একটাতে তো থাকছেই তোমার তুমি যদি প্রাইমারটা অ্যাটাচ করে রাখছো প্রাইমারের যে এরিয়াটা সেই জায়গাটা তো আরএনটিপি ওটাকে ডিগ্রেড যদি না করি রেখে দিই তাহলে ইউগুলো রয়ে যাচ্ছে এবার তোমার কমপ্লিমেন্টারিটা ইউ থাকবে না কিন্তু যেটা ইনিশিয়াল যে স্ট্যান্ডটা তৈরি হলো সেটা তো ইউ রয়ে গেল তাই না তো সেই ইউটা ওই ইউটা আমি রাখতে ওই এনটিপিটা রাখা যাবে না ওটাকে ডিএনটিপি করতে হবে কাজেই ওটাকে ডিলিট করে আবার ওখানে নতুন নিউক্লিওটাইড ডিঅক্সিরাইবোজ নিউক্লিওটাইড অ্যাড করে আবার লাইগেট করতে হবে So here you will see the important feature of DNA polymerase 1. You see the DNA polymerase 1 is it contains two different subunits. So when we treat it with some proteolytic enzyme, the proteolysis, because it's a protein, DNA polymerase 1, the proteolysis of polymerase gives rise to two fragments, one larger fragment and one small fragment. Okay? When we do that extracellularly on the outside of a cell, we can do this outside of a cell in a environment outside in vitro process, right? We add some proteolytic enzyme, it degrades the polymerase 1 hollow enzyme. Hollow means when the enzyme is filled with multiple subunits. So they separate into two fragments, one large, one small. The large fragment is known as Klinov fragment and the small fragment is basically having 5' to 3' exonuclease activity. Now this Klinov fragment contains two fragments, here you can see green and yellow. One with the 5' to 3' polymerization activity, another one is 5', another 3' to 5' exonuclease activity. Means basically all the other Pol 2, Pol 3 that they share 3 to 5' exonuclease activity which is also being shared by Pol 1. So 5' to 3' polymerization activity, 3' to 5' exonuclease activity. This is present in all polymerase 1, 2, 3. But the 5' to 3' exonuclease activity is rare. It's found in only Pol 1. Okay? Only Pol 1. And the moment we use the proteolysis, we have these two fragments, one large, one small. So the large fragment is capable of carrying out the function of both 5' to 3' polymerization activity, 3' to 5' exonuclease activity. And the rest the 5' to 3' exonuclease activity gets separated, and this is not always needed because if you want to delete something or delete added nucleotide, we need to go backward. That is 3' to 5'. That's why always the exonuclease activities are present 3 to 5' and not 5 to 3'. Now we are going to see the examples of eukaryotic polymerase and look at the comparison between them. Here you can see the eukaryotic polymerase. The polymerase in eukaryotes are named as alpha, delta, epsilon, beta, gamma. You can see the delta and epsilon are known as the predominant polymerases in eukaryotes. They help in elongation of leading and lagging strand. Epsilon involved with the leading strand, delta with the lagging strand. On the other hand, polymerase alpha is simply used for the priming of replication in both the strands. And polymerase beta involved in DNA repair, polymerase gamma in the replication of mitochondrial DNA. Because in eukaryotes, organelles like mitochondria have their own DNA, and the mitochondrial DNA replication is done by polymerase gamma. Alright, so these are the different types, different DNA polymerases, their classification and comparison.
So for the synthesis of leading and lagging strand in case of prokaryotes, there are no.
No, same pol 3. Now we are going to talk about the DNA polymerase structure, how they look like. This is the DNA polymerase 3 or pol 3 structure. Basically, this is a overall idea of DNA pol 3. There are different sub assemblies to a pol 3 structure. You can see it's made with multiple different subunits. So it looks something like two arms, two hands connected to a body. So consider this as a body known as tau unit holding two different mirror or identical sides of the polymerase together. And at the bottom, let's say this is a leg which is built with gamma, delta, chi, psi and all these different structures. Now these three different portions, the body, main body, the arms made up with alpha, and this is known as delta, epsilon, theta, and there is beta clamp which will be loaded here. Okay, so if you consider this as an overall structure, beta clamp has an important specific role to play, tau has a role to hold them together, and this leg which is made with delta, psi and all this segment, they help to load the DNA onto the beta clamp or beta clamp on top of the DNA. Basically, imagine this is the DNA strand. The DNA strand will be separated, right? Once it's separated, the polymerase will load itself. So let's say this is the polymerase alpha units, tau connected, okay, alpha units the body, and this is the loader. The beta clamp will help to load the polymerase on top of both the template strand, because the DNA replication process that is going on in the prokaryotes, it's going on in both the strands, both strands simultaneously. So the beta clamp is loaded in both the strands and the polymerase will move from 5' to 3' direction of newly synthesizing strands direction. So this was, let's say, the red one is the existing parental 3' to 5'. So the new strand is the blue color 5' to 3'. This is new, and polymerase's movement in this direction, right? So the role of beta clamp is to load the polymerase, and the alpha units and tau hold the structure together, and the rest, these are known as the clamp loader, and they are also known as the clamp loader means they load this beta clamp on top of the hands, you can say, the yeah. Not tau, the hand means this epsilon, this structure. So they load it there, this area, beta clamp loader. So what is the point of having beta clamp? It looks like a clamp. What is the function of a clamp? Is to hold something, right? So there are two different strands of the DNA here, you can see in this picture. And with the help of the beta clamp, the polymerase is holding itself on top of the DNA strands. So it's something like trapezium, the trapezoid, that game, you know, they have this rollers that hold them in two ropes. For example, two ropes are passing, and you have clamp loader. With the help of the clamp loader, you are holding in both these ropes together, right? Now you will be moving in the one forward direction, that is 5' to 3' of newly synthesizing strand. Without the beta strand, the polymerase can polymerize, but the rate of polymerization will be very, very low. Okay. So what is polymerization rate? Number of nucleotides added per minute or per second, that is polymerization rate. And what is processivity. There are two things. One is polymerization and the second one is processivity. What are the difference between the two? Polymerization means number of nucleotide per time, per unit time. That is polymerization rate. What is processivity? Number of nucleotide added before the pol falls off. Number of nucleotides a polymerase can attach in one go, in one stretch. That is known as the processivity of that DNA polymerase. The processivity of DNA polymerase 3 is maximum. That's why DNA polymerase 3 is used as a primary polymerase for DNA replication in prokaryotes. That is maximum for delta in case of eukaryotes. So delta and the epsilon, because that's why they are used for leading and lagging strand. Particularly for polymerase, it's in prokaryotes pol 3 that's working. Now without the beta clamp, the processivity drops. Polymerase can polymerize, but let's say 10, 20 nucleotides and the polymerase falls off. But with beta clamps loaded, with beta clamps tagged, it can polymerize 1000 nucleotides at a time before it falls off. Now the rate of polymerization depends on the availability of DNTPs. But processivity is not related to the availability of DNTPs. It is also, but processivity depends on both the factors, the availability of DNTP as well as the presence of beta clamp. Okay, while polymerization only depends on the DNTPs, the amount of DNTPs. If there is no DNTP, no polymerization. In both the case that's true. But in case of processivity, DNTP along with DNTP, beta clamp is another important parameter. In this picture you will see a schematic drawing of polymerase, DNA polymerase 3, the similar drawing you can see. So you don't need to remember it, only need to know the importance of tau unit to hold two identical structures left and right hand side together as a single polymerase body. Beta clamps are very, very important in order to load the polymerase itself to the single strands template DNA. And what else? This gamma and psi, this structure is the loader of beta clamp. They load beta clamp on top of the strands of the DNA. That's all you need to know. Okay. Now one big question comes in, that we know that this polymerase, two different strands are present in the DNA. This is strand 1, this is strand 2, we are seeing two different strands. And these strands both are used as a template strand to synthesize a new strand. Right. Now let's look at the dilemma here. We have this 3' to 5' existing strand, and we have 5' to 3' existing strand. So our new strand synthesis will go this direction, 5 to 3', and this is also 5 to 3'. This polymerase both are connected together by tau. So polymerase at a time can move at only one direction, either this or this. If polymerase move right hand side, it's moving 5 to 3' for this strand, but for this it's opposite. And if polymerase is moving from this 5' to 3', this is fine, but for this strand it's opposite. If they are not connected by tau unit, two are separate units, they could have easily polymerized two strands separately. But they are moving in opposite direction. Let's assume these are the two beta rings. They are moving in opposite direction, but they are connected together. How they move? There should be shearing force and they should separate. So to prevent that, there is a fold. Formation of loop is done. So that concept of DNA looping, we'll see that in a moment. So DNA is bent in such a way that at a time the polymerase will move only in one direction. Of course, one of the strand will be smoothly replicated, and the strand with smooth continuous replication will be known as leading strand. and the one with breaking small segment replication at a time will be known as lagging strand. Okay. Now this bend is important for the replication to continue even when the polymerase are connected. The two units, two sides of the polymerase are connected together.